So in this video, this is our transcription and post-translational processing part 2. So you should have already watched part 1 as well as the HHMI video. So in this case, we are going to continue our discussion. So we have made it through these 2 items are covered in video 1. And so right now we're going to talk about transcription in prokaryotic cells. And so we're going to begin with initiation, talk about elongation, termination, and then we'll continue our discussion. So initiation, this discusses how this all begins. So the closed complex is formed upon the recognition of the promoter. So if you remember, our promoter is composed of 2 regions, the -10 and the -35. So the sigma subunit begins to combine, it identifies a promoter, and then it begins to assemble the complex. So the closed complex indicates that the DNA is not separated. RNA must be copied from the antisense strand, or we're going to use that as a template so that the RNA fragment is read in the sense direction. So the initiation is before it opens. So we're going to go from closed to open. And so this here is a picture of the open complex. And we can identify that by this region right here where we see an opening in the DNA. And that opening is going to be 18 base pairs. So the initiation starts and stops, usually releasing a small RNA fragment. So you can think about this kind of like a stutter. It starts and then it's like, I'm not quite ready for that. And then it'll do this over and over again. And so it releases RNA fragments on the order of 5 to 8 base pairs or bases. Those then are frankly degraded and depart. The DNA unwinding is still completed by a topoisomerase. So topoisomerases unwind and relieve the torsional strain. So initiation. The sigma subunit dissociates after 10 bases are added to the strand. So on the previous slide, we talked about how the complex starts and stops, releasing between 5 and 8 bases. So the sigma subunit remains bound until we get past 10 bases. Once we get past 10 bases, it is going to depart. These stutter steps, that got exciting. The stutter steps seen here, the sigma subunit is still bound. So that's important to note that the stuttering does not lead to the deportation or to the departure of the sigma subunit. And so the remaining complex is very stable and remains together until termination. So once we move past the 5 to 8 base pairs, we get all the way to 10 and the sigma subunit departs. This complex is going to stay together. So, and this complex will continue to add RNA bases over and over again. So RNA polymerase is considered open when the DNA is unwound, approximately 18 bases. The sigma subunit promoter leaves after the first few bases are polymerized. NUS-A, which you can see over here in this image here. So we've had the departure of so this light blue similar is the sigma subunit. It will depart and the NUS-A will come in. In this image, we can see that they're in the exact same location, and that's not necessarily true. And so NUS-A adds to the holoenzyme complex after initiation during elongation. So sometime between 10 RNA bases and termination, we're going to add NUS-A. So initiation really terminates in the departure of the sigma subunit. Elongation covers everything in the middle. So RNA elongation is bases being added onto the 5' or the 3' end, of the RNA strand. So in our open complex, which we can see here, We have our 18 base pair bubble, which is right here. And so this is consistently open. And as the RNA is created, as the complex moves down the DNA, the template is going to turn back into double-stranded DNA. So it is unwound, copied, and then rewound. So at no point other than during the open complex are DNA bases exposed to the environment. And so we can see in this graphic over here, which we will zoom in on, we have translocation, which is similar to the ratcheting that we will observe in the protein translation in the next chapter. But until then, what we see is that we add on a base here, and then we are going to continue to add over and over again, and this cycle repeats. So elongation is a repetitive polymerization. So this repetitive polymerization is the same step over and over and over again. So if we look at this arrow pushing mechanism, which hopefully looks very, very, very similar, the main difference, if we use our highlighter, is this. So the big difference here is that we've added a 2' hydroxyl. The way in which the electrons move from the 3' end to the incoming base is the exact same. It still requires 2 magnesiums that we can observe over here. So we still have 2 Mg2+, as well as a variety of residues. And we're going to have the departure of the phosphorus leaving group, which encompasses the beta and gamma strands. And so we can see this whether we look at the reaction here that shows the template. So this, keep in mind, is a DNA template. And we are creating RNA. And so bases are added based on their ability to create a Watson and Crick base pair with the template strand. So the Watson and Crick base pairs that we learned previously really control how all of these different parts interact with one another. So backtracking. What happens if we make a mistake? We know that there's no sorting because there's no way for the base to know prior to arrival into the strand. So if there was sorting, the error rates of about 1 to a million would make sense. But there's no sorting. There's also not a exonuclease somewhere else. So if you remember back in DNA replication, it has to unwind 8 bases in order for you to cleave off whatever is on the end. In this case, we see some slippage where it will backtrack and slightly unwind and then move forward. So in this case, we mostly see this when we have a mismatch destabilizing the complex, allowing it to move backwards. You can think about this, since it can't create the hydrogen bonds that we know stabilize DNA and RNA, it basically unwinds because it can't really be solid enough to move forward. And so once it moves backwards, it doesn't contain a discrete exonuclease, meaning the bases are not removed from a different location. The bases are removed in the same active site, which is different than DNA polymerization, where the exonuclease is far removed from the polymerization site. So elongation is pretty simple. It is the repetitive polymerization of adding bases. However, at the end of the gene, it's got to stop because our mRNA fragments don't necessarily need to be quite long. They should really just encode for the gene. So at the end of the gene, there's a couple of different types of termination sequences. So we have our ATG start codon. There are stop codons, which, if you've taken genetics, are things like TAA or TAG. And so this termination sequence here is different than the stop codons. So your mRNA fragment has a leader end between the promoters and the start codon and a lagging end, or an end, we'll call it the 3' end. There's extra stuff on the end. So this termination could include a palindromic sequence that's going to increase pausing, or it's going to create a hairpin. So the gene itself, the protein of interest or the cellular machinery that we are interested in, is found in this region here. And so in this case, we're looking at the excess parts of mRNA. So it turns out that for the purposes of DNA or RNA termination in bacteria, there are other ways that this happens in eukaryotic cells, but right now we're just talking about prokaryotic cells. So we have row dependent and row independent. And so both methods involve a hairpin, and so we learned about hairpins, and so we can see this hairpin over here. And so this hairpin is going to destabilize the duplex of DNA. NUS-A that we met a few minutes ago, NUS-A shows up after the sigma subunit departs. NUS-A increases pausing. So you can think about this as a protein that is like, you've made a hairpin. Don't you want to hang out here for a little longer? So the increasing of pausing on the order of 10 to 30 minutes, it basically stops or stalls transcription, and it resumes when the hairpin melts. But the longer lifetime of the hairpin can induce termination. So first we're going to talk about row independent. So row is a protein. So this is termination without an assistive aid. At the end of the gene or after the stop codon, there's often a GC-rich sequence that's going to result in a stem loop structure upstream of a poly-U region or just post-stop codon. So this is another word for a hairpin. And so if you remember, they look something like this. And so we've created this in our RNA. And the stem loop structure in the transcript terminates synthesis. Basically, it stalls so long that it is then going to dissolve the polymerization complex. And so when this happens, we have the removal of our stem loop structure here. We then have the poly-U tail or a poly-A tail. In eukaryotic cells, that is added in a different manner. And so what we see in row independent is that a hairpin forms. Eventually, the holoenzyme complex dissociates. And once it dissociates, we can then remove that, or it terminates in such a way that the mRNA fragment separates from the DNA. So row dependent termination is, in my opinion, slightly more interesting. Now, row rapidly invades the elongation complex and then slowly dissociates. So if you remember, the sigma subunit has left, NUS-A has come in, and it's encouraging the pausing at a hairpin. So the elongation complex is just moving along. So the rapid trapping of the elongation complex is important to establish termination. 80 RNA nucleotides wrap around the row complex, which is, as we see over here, this is our row protein, and it is a hexamer. Just a reminder, hexamers are 6 subunit proteins, and in this case, we will come to see that they are 6 identical subunits. So 80 nucleotides wrap around the row complex. The row complex then helically twists, so it basically rotates, forcing the RNA to dissociate from the DNA. This occurs when single-stranded RNA is formed after the coding region. So we have the gene, we're in the excess region afterwards, so we've had roughly 80 nucleotides that will wrap around row in the row recognition site. Row then essentially rotates, so row rotates in this direction per this graphic, and it essentially, in my opinion, yanks the mRNA fragment out of the elongation complex. So once we remove it, the whole thing falls apart. And now this is terminated. And so what we see is that we have the ability to continue to remove how all of this works. So in row-dependent termination, we can see our 6 subunit row here and here. And so the way the mRNA fragments bind in here, then we have this helical twisting. And so what we see is a rotation around the center point. And so when we see this rotation in this direction, it is going to essentially remove the mRNA from the elongation complex. So termination involves the formation of a hairpin. The hairpin can induce binding of row, which then twists and removes the mRNA fragment or any RNA fragment. Or we can have row independent, where a hairpin forms, forcing the collapse of the entire elongation complex. So as a reminder, the transcription overview, we have DNA that's transcribed into mRNA or other RNA fragments, such as rRNA or tRNA. And so transcription is initiated when the sigma subunit binds. After roughly 10 to 12 bases, the sigma subunit will depart, and we've moved into the elongation phase. At some point in the elongation phase, NUS-A will bind. When NUS-A binds, we can then move forward. Eventually, NUS-A encourages pausing. It just informs that that's a good idea. So the increased pausing can lead to either row-dependent or row-independent termination of transcription. So turnover. When we think about mRNA turnover, when we think about the word turnover, it means to be regenerated, basically to go from start to finish over and over again. So in bacteria, transcription and translation can occur simultaneously. So the transcription machinery will move past the 5' end of the gene, and then the translation, which is the protein production machinery, will bind to the mRNA fragment, and those two entities will move along together. Most mRNA in bacteria have a half-life on the order of 2 to 3 minutes. So by the time mRNA is started to the end, the mRNA starts to get degraded quite quickly. So mRNA can be degraded in the degradosome, which is a multi-protein complex that contains nuclease, RNase E, and an RNA helicase, and a phosphorylase. This degradation starts on the 5' end. So this is different than your traditional RNA degradation. We'll call it acid hydrolysis. So this is the enzymatic degradation, and that starts on the 5' end of the mRNA fragment. The acid hydrolysis can occur anywhere. So I think the best thing is to stop this video here, and then we will pick up in video 3 with the post-translational modification of mRNA or any RNA fragments and the transcription in eukaryotic cells.